Alright, so welcome back. Today we're here for part 2 of our chromatography lab. So just to refresh your memory, our scenario was we found some dead fish in a river near our school. And we did some internet research and we found out there's a toxin called rafinotoxin that has a lot of the same symptoms as what we're seeing with the fish in our river. And we were looking for different possible sources for a rafinotoxin, and we found a company called Acme Widget Company that was creating a waste. And here was a sample of the waste product that we collected, and they said that there was no rafinotoxin in here. As you can see, this has a green color, whereas our rafinotoxin has a red color. We still decided that we wanted to test this to see if there was actually any rafinotoxin present here, and we decided to use some chromatography techniques. So again, chromatography separates substances into their individual components, and there are many different factors that we can separate by. So we can separate by size, we can separate by charge, or we can separate by how hydrophobic the different molecules are. In part 1, we did gel filtration chromatography, where we ran this substance through gel filtration, which looks like this. And we separated our components of our waste by size. So we separated by size. And we have 3 different components so far that we have found in our waste product. The first thing that came out was our blue substance. Since our blue substance came out first, this is going to be the largest of the 3 that we collected. The middle substance that we collected, gotta get it out. The middle substance that we collected was brown in color. We put this one in a special little tube because we figured it's brown, there could be rafinotoxin in here because we know rafinotoxin is red, so we're going to further test this today. The third substance that came out is our smallest. We know that because the smallest molecules come out of our chromatography filter last. The last thing that we collected was this yellow substance. So by size, we were able to separate our waste product into 3. We had, again, our red, our brown, and our yellow that came out of here. Again, blue came out first, so we knew it was the largest. The yellow came out last, so it was the smallest. But we do know one thing about gel filtration is that if 2 molecules are very close in size, they can't properly be separated. So while we were able to filter this out into 3 components, there could be multiple components mixed together in any of the samples. And our goal is to look for the red rafinotoxin, and of the 3 that we have, the one that looks closest to something that could contain a red toxin is the brown, and that's why we collected it separately. So what we're going to do today is a second type of chromatography, an effort to separate this out and see if there is any red rafinotoxin present in that sample. These other ones I'm going to go ahead and discard because I know that there's no red rafinotoxin present in them. So what we're going to be doing here today, the last question in our lab from last time asked, what other properties do we know about rafinotoxin that we could use to help isolate it from our sample? So again, here's our sample that we're going to work with, and this is what we got from part 1 of our experiment. So one other piece of information that we had from our part 1 scenario, and if you want to pull that back out, we're going to look at it here for a second. Another thing that we know about rafinotoxin, other than its size, it told us that its isoelectric point is 7. And again, an isoelectric point means that well, an isoelectric point of 7 means that at a pH lower than 7, the charge on the toxin is positive. At a pH higher than 7, the charge on our toxin would be negative. So what we're going to do today is a process known as ion exchange chromatography, and you all know ions. Ions are molecules that have either gained or lost an electron and therefore are charged. So what we're going to do is separate any components of this substance by charge. And the way that we're going to do that is by controlling the pH in the column. So here is our ion exchange column here. And what we are working with in our ion exchange column is we're going to be separating proteins based on charge. Ion exchange is typically used to separate proteins based on their charge by utilizing their isoelectric point. So again, for a phenotoxin, pH lower than 7, it's positive. pH higher than 7, it's negative. So we're going to use what we know, which is how it's charged at different pHs, to help us separate this in its components by using pH and the pH of the substance that we filter through the column. So essentially what's happening here is inside of this column here where the gel part is at the bottom, we have small beads, and these beads have a fixed charge. In this case, the charge on them is negative. So what we're going to do is slowly change the pH of the substance that we filter through, and as we change the pH, the different substances here are going to bind differently. Initially, when we wash through, we are going to be separating components out. So our negative molecules are going to bind to the beads because the beads inside are negative. So anything in here that is negative will bind. And then we will wash it with a stronger positively charged substance. And when we increase the positively charged substance that we are flushing through, then our negative thing is going to filter out. So essentially what we are doing is changing the strength of the liquid we are running through the column to separate these molecules by charge. So again, I want to be safe as we do this. So put on my gloves. Alright, so our first step, let me turn you here. So again, I have my nifty little setup here, and I went ahead and I put a piece of white paper behind it so that hopefully you can see colors as we work here today. So the first thing I want to do is place a beaker under it so I can catch the thing. I'm going to open the bottom and the top, and as we remember, these were tricky to get off. There we go. There we go. So there's the first one. And then the bottom. And it'll start dripping. So I'm gonna, step 1 says let the buffer go ahead and elute. Elute means drip or run through until it stops dripping. Alright, so what I'm going to go ahead and do, I'm going to add some more buffer here, and the reason I'm doing that is because I want to wash this out to ensure that there's nothing inside that's going to contaminate my results. So I'm going to add some more buffer there, and I'm just letting it wash through. Again, the goal is that this is going to wash out anything that may be residual from anyone who used this column before me. So I'm washing it out, and you guys can kind of see it dripping there when I move the paper back. Hold that up when we get our colors there. But right now, it's dripping through. Again, I'm doing this so that I'm cleaning out anything that may still be residual inside. So again, as we work on ion exchange chromatography, we are separating molecules, or in this case proteins, based on their charges. So inside of our filter, or inside of our chromatography tube here, the matrix with the beads have a negative charge. So anything that's positive is going to initially stick to the beads, whereas the negatively charged molecules are going to go ahead and wash through. So remember, opposites attract in this case. So anything negative is going to go ahead and bind to the bead. The beads are negative. Anything positive is going to bind to them. And the negative thing is going to come out first. But then as I change the pH of what I'm washing through into something stronger, I'm going to wash through something positive, and then the beads are going to, or the initial negative molecules that stuck to the beads are going to switch and start sticking to the positive thing I'm washing through. All right, so we just stopped dripping. So it says place a new test tube under the column. So I'm going to get me an empty test tube. And I am going to add this substance that we had from experiment 1, my brown substance. So I'm not going to inject all of it. I'm going to take about half of it or so here. And the reason I'm doing that is so that in case something happens and I mess up, I still have some left. Because we got plenty here to work with. Alright. So, and if it separates, great, then I can add the rest of it. But I'm going to just start with this here. I'm going to let it separate. So again, inside here, our beads are charged negative. So as this liquid goes through, anything that's positive will stick to the negative beads. The opposites attract. So the negative beads will stick to any positive protein inside. And then what I am catching here are the negative substances, because the negative substances are going to wash right through. So anything that I am currently catching is negatively charged. And I will hold it up here as soon as it stops dripping to let you see the color. This is a very bright, almost clearish yellow substance that I'm catching here. So this is brightish yellow substance, and we can start seeing some color collecting at the top. So it looks like this is separating out really well. So I'm going to go ahead and put in the rest of our toxin, or possible toxin. So it's our rest of our brown substance. I'm going to let the rest of it go through. So again, our beads are charged negative. Anything positive will stick to those negative beads. Anything negative is coming right through, and that's what I'm collecting in my test tube. So right now, I am collecting any negatively charged protein. And as I filter more and more, I can start to see a bright red color forming at the top of my column. So for some reason, it's not quite as bright red on the screen there. But this is red at the top. And what I've collected so far, again here, is yellows. Look at the stark contrast. So I already know that that unknown that we had collected that was brown from part 1, I know it has 2 components. I have a yellow component and I have a red component. The red component is obviously positively charged because it's stuck to my beads. This is negatively charged. So I'm going to set this aside. I'm going to go to the next step. So it says wash the column twice with a half a milliliter of loading buffer to let it continue to wash through. So what I'm going to do, put my beaker back underneath, and I'm going to inject roughly 1 total milliliter of my loading buffer. And again, this does not have a strong pH, so I'm just rinsing out. Right now I'm still rinsing out anything that is charged negative. So I am not yet collecting my positively charged red. This is just rinsing out anything else that is negatively charged. Okay. Step 4 says go ahead and continue to wash until what comes out looks colorless and is only concentrated at the top. And I'll put one more pipette full through here, but it's already starting to look fairly colorless. But just to be sure, I'm going to go ahead and continue to rinse it just a little bit longer. One thing about these ion columns is they do kind of go fairly quickly, so I'm just going to let this rinse through and we'll move on to step 5. So what I'm going to do in step 5, I am going to, so if this is our ruffinotoxin, what we know about it is it's positively charged when it's in an acidic environment. So right now it's in an acidic environment and it is charged positive. Since it's charged positive, it sticks to our positive beads. It sticks to our negative beads. It's charged positive, it sticks to the negative beads, and that's why it's not coming out. Because right now, the substance that I'm flushing through at the top is an acid. But as soon as this finishes dripping through, I am going to change the substance that I am injecting at the top, and it's going to be a base. So I'm going to start injecting a base. And what happens, again, because of the isoelectric point of ruffinotoxin, when it's in a basic environment, the charge of the toxin changes to negative. If the charge of the toxin changes to negative, it's no longer going to stick to these negative beads. It's going to wash through. So what I'm going to do now, I'm going to move this out and I'm going to get me another clean beaker or test tube. So here's my clean test tube. And again, this next substance is a different one. I haven't put this in yet. This is a basic solution. When ruffinotoxin, I'm assuming this is ruffinotoxin because it's red in color. When ruffinotoxin is in a basic environment or has a basic solution, sorry, my thing died here. So when it's in a basic environment or when I push through a basic solution, the toxin will change charge to negative. And if the beads inside are negative and the toxin is negative, it's going to wash through. It's no longer going to stick. So this should happen fairly quickly as I put these in. All right, so if I look, you see my red is starting to wash down. So it's washing down. So what happened? I put in the base. When I put in the base, my toxin changed charge. Now it's negatively charged because the toxin's isoelectric point is 7, meaning as long as I was putting through acid, the charge on the toxin was positive. And when it's positive, it's going to stick to negative beads. But now that I'm putting through a base, when the toxin is in a basic environment, the charge it has is negative. If it's negative, it can't stick to the negative beads, meaning it's going to come straight through. So I'm going to continue to add this in hopes that I'm going to wash out the rest of my toxin. So as I can see, my toxin is coming out, and I'm collecting it in this tube. And we clearly saw, this is going to be a confirmation for us, that this is ruffinotoxin because one, it was very, very dark red in color. Now that I've mixed through this basic solution, it's going to be a little bit lighter just because now I'm mixing that concentrated red with a liquid. So it's going to be a bit more diluted, but I'll hold it up here in just a second and you can see that the liquid I'm collecting is red. And we also confirmed that this is ruffinotoxin because it's stuck to the beads when I washed through an acid. But when I washed through a base, it flowed through, meaning we confirmed its isoelectric point. So here, this is red. It might be light, but again, I did mix it with some liquid here. So this is red, meaning this is ruffinotoxin. So this is the ruffinotoxin. Now, I'm going to just set this aside. I'm going to set my whole little setup aside. And I am going to rinse it through because, again, I want to be sure that nothing in here is contaminated for the next person who uses it. So I'm going to go ahead and just let that drip over to the side there. And I'm going to turn you back around. So essentially, what we did in parts 1 and 2, we had an unknown substance leaking from the Acme widget company. They said this did not contain ruffinotoxin to kill our fish. But what we did in lab 1, we used gel filtration chromatography to separate the mixture into 3 components. We had the larger blue protein, we had a brown protein. Here's a little bit of it left here of the brown protein we got. And we also collected a yellow protein using gel filtration chromatography where we separated these out by size. However, we knew that the ruffinotoxin was red in color, and we also knew that if there were proteins in there similar to it in size, they would come out together, which could contribute to that sample being brown. So we took this brown sample and we used ion exchange chromatography to separate these proteins by their charge. And we did that because we utilized what we knew about the isoelectric point of the ruffinotoxin. We knew that in an acidic environment it was positive, but in a basic environment it was negative. So what we first did is we washed it with an acid, and in that we collected this negatively charged, yellowish golden colored protein. But then when we washed it with a base, we got this red substance. This is going to be the ruffinotoxin. So in total, our initial sample contained 4 things. We separated out 3. In part 1, we separated out 2. 2 things were in that 1 from part 1. So we had 4 total substances that we separated out, one of which was ruffinotoxin. So we can officially say that the Acme Widget Company was producing ruffinotoxin, which was killing the fish. And that would allow us to talk to them and or bring up a lawsuit against them if needed. So for the rest of this, make sure you go ahead and you answer your questions. So it asks at the end of experiment 2, describe your results. Did the brown fraction contain more than one component? So did we get out multiple things from our brown fraction? How do you know that the brown and the red components are not the same thing? So using what we did today, how do you know that the brown that we had at the beginning and the red we had at the end weren't the same? How do we know those weren't the same thing? Question 3 asks, since the red protein bound to the negatively charged ion exchange resin at an acidic pH of 6, what is the charge of the red protein at pH of 6? Okay, next question. Do your results support the hypothesis that the Acme Widgets is releasing ruffinotoxin into their wastewater? So do we think that the Acme Widget Company is releasing ruffinotoxin? And then in what way did your results support that? So how can we actually confirm that the Acme Widget Company is releasing the toxin? And then as a final summary here, it says, so there is evidence that Acme is releasing chemical that has the same characteristic size and color as ruffinotoxin. The fish kills with the symptoms of the ruffinotoxin are certainly evidence as well. At this point, you'll need to call in the experts for more tests and to confirm that the red chemical that you have is actually ruffinotoxin. What other experiments might the experts perform to prove or disprove the presence of the ruffinotoxin? So what else could be done by professionals to figure out what is actually being released here? So that is the end of this chromatography. We have one more coming up that's more of a fun one just to show you something from your everyday life that we can use this for. So that is it for now. Make sure you answer your questions.